that the two tools you've learned so far, once DNA is extracted, are restriction enzymes to cut the DNA up into more manageable pieces. We talked about two different restriction enzymes used for different things. Restriction enzymes that make straight cuts, like kind 2, are fine for the next thing you're going to learn, like DNA fingerprinting. Fine if you don't care that the DNA can't recombine with other DNA. If you want that, you need a restriction enzyme like EcoR1 that leaves crooked cuts, which any two pieces cut by that same enzyme can recombine, which opens the door to putting genes from one organism into another organism, which we call transgenic organisms. So the key is those sticky ends. Then gel electrophoresis. The idea that, and I've rearranged this slide a little bit, that's why it looks weird. The idea that once you've cut the DNA up, you need a way to sort it. And the way we sort it is by size. We pour a little block of Jell-O. We make little wells in it. We put the mixed DNA fragments in the wells. We run a current, which works because DNA is negatively charged. It'll get dragged towards the positive terminal through the gel, and the little pieces will go faster and farther than the big pieces. So they sort themselves out. One of the things, and I don't remember if I mentioned this yesterday or not, one of the things that's confusing to students about this is, well, okay, how is it you're saying these pieces are different sizes, but all of these pieces are the same size? Look at them, the bands are all the same size. Here's the best explanation I can give you for that. They are that size and shape because that's the size and shape of the wells they were put in. What you're seeing is that we put a bunch of mixed fragments in this well, and then we dragged them this way. Well, the smallest fragments went the farthest, right? But all those little pieces, there's a whole bunch of the same piece, right? Because you had a whole bunch of copies of that organism's DNA. Well, they all went the farthest, but they're still kind of roughly in the size and shape of the well you put them in. So that band is there. The bigger pieces, you know, this piece was a little longer, well, there's a whole bunch of it too. And those pieces went this far. And so they're still in that size and shape. Well, it's bigger, but it's not supposed to be. You see what I mean? Like, it's not just one piece. It's a whole bunch of the same length of piece that went that distance. Clear? Questions? I think that's the thing that confuses students the most. Okay, so then you got to stain the gel, but then you have this kind of picture of, okay, how big were the pieces and how many of them were there? And is one of those pieces what I want? There's ways to figure that out as well. So one of the applications of this I want to show you now. Is DNA fingerprinting. After yesterday's activity, it's possible you already kind of get this, but just in case, I want to lay it out. You know, let's say I think that Eli killed somebody. Somebody got killed, and at the scene, we found some blood. There was some blood at the crime scene. I think that blood belongs not to the victim, but to Eli. I want to analyze that blood and put Eli at the crime scene, right? I want to establish that he was there by identifying that blood as his. Restriction enzymes and electrophoresis are how I do that, right? Basically, I got to get some of the cells from that blood. I got to get some cells from Eli, which is pretty easy. You leave cells everywhere. All of you do, right? On the lip of a glass you were drinking out of, just a piece of his hair, whatever. Need some of his cells. Probably want some of the victim's cells as well, just so I can make sure the blood didn't come from the victim. And then I got to do the soap, salt, alcohol trick of extracting the DNA, right? But once I do, it's just, okay, and they sell specific packs of restriction enzyme. There's like a DNA fingerprinting pack that has just the right enzymes to cut up anyone's DNA into a bunch of different sized pieces, but slightly differently for different people. Because we're all slightly different, right? I mean, you're 99.9% the same. Your DNA is, but that 0.1% is enough. All those little differences stretched across your genome mean that the pack of restriction enzymes used for this is going to cut everybody's DNA a little bit differently. So victim, blood, Eli. And now I'm just looking for, all right, the blood gives me a characteristic pattern of bands. If either the victim or Eli's bands are exactly where the blood bands are, they're from the same person, right? So if Eli's bands match the blood's bands, that puts him at the scene. If it matches the victim's bands, yeah, try again. If it doesn't match either of them, ooh, maybe we can find somebody else, right? So now I chase down Iola, get some of his cells, see if that matches. That's the idea, okay? So you compare those bands to identify different people. In the same way, you can identify family relationships. The closer your bands are to someone else's, the more closely related you are, the more similar your DNA is. So paternity testing works like this, right? Family relationship type testing. So I wanted you to see that application of it. Okay, the second to last tool we'll need to get to glow in the dark cat is called PCR, or polymerase chain reaction. Basically, the problem is a lot of times we'll find a piece of DNA we like. We need more of it. We need more of that piece to have enough to work with. Or we'll find some of the DNA we want to analyze, maybe that blood at the crime scene. There's really not enough of it to run on electrophoresis. I need more copies of that DNA I found at the crime scene. That's PCR. It's basically a DNA copy machine. It's artificial DNA replication. That picture could be better. I don't love it. But yeah, this is kind of just like a DNA copy machine, except it looks a lot different than I thought it did when I first saw it done. I was kind of surprised at how simple it is. You basically, you have the DNA that you like in little containers like this, just little plastic tubes. And into that tube, you add a couple of things. Most importantly, what you add is DNA polymerase molecules. Remember DNA polymerase? That's the one that in DNA replication adds the bases in opposite directions. Remember that? You add a special version of that into these tubes. But then what you end up doing is just putting all these tubes in like a tray into a water bath that's in this machine, like about this size. And then you close the lid on the machine and you press a button and you just walk away from it. And you come back like 3 hours later and you have millions of copies of that length of DNA that you put in this tube. And the reason it works, this is so weird. It turns out what they couldn't figure out was how do we make the helicase do its work? Like how do we do artificial helicase? How do we get the DNA string to split so that it gets copied and then come back together and then split again so it gets copied and come back together? They couldn't do it until somebody figured out, hey, if you just heat the DNA, it makes the weak hydrogen bond split. Now we just need a polymerase that'll work at that high of a temperature. You know where they got it? Yeah, they got it from the bacteria that live at the bottom of hot springs in Yellowstone National Park. Crazy. So basically, you heat the pieces to unzip them, and then the DNA polymerase molecules that they found in Yellowstone do the base pairing. And all that water bath is doing all day is heating up and cooling down and heating up and cooling down. And every time it heats up, Yellowstone polymerase runs along and copies it. It cools down, it comes back together, heats up again, splits all of them. So you're just doubling the amount of DNA every time. We are almost there. We are almost to glow-in-the-dark cat. The only problem we have, go ahead. No, it's that, well, yeah, if they're separated because of the heat and you got the polymerase working, if you keep it that hot, yeah, they'll fall apart again. They'll dissociate again. Yep. And I think there's a timing that matters too. Like you only keep it hot for long enough to get the copying done, and then it starts to cool back down. The problem with glow-in-the-dark cat that we have right now is, okay, you know, you use the EcoR1 to get the firefly gene out of the firefly, right? And then you take a cat cell and you cut the DNA open and you put the firefly DNA in there, you get it to stick in there after you've done the PCR with it and you've sorted stuff. Uh-oh, might be about to get yelled at. Hello? Hey. Yeah, we do. Yep. No problem. Right. There's extra stuff there, right? When we're done with notes, would you mind running those down the clock? All right. The problem we have is that when you stick the firefly DNA in the cat's genome, you've only done that in one cell. It's not going to light up the cat. It's just going to light up one cell of the cat. And you're not going to be able to see that. That's no fun. So what do you got to do? Anybody know? You got to clone the cat. You got to basically take that one cell that you put the firefly gene into and make it turn into a whole cat that has the glowing gene in every cell in its body. You got to clone it. So that's our last technique. Now, technically, cloning is just producing an organism that's genetically identical to another organism. Technically. Is that something that can happen via sexual reproduction? Why shouldn't it be something that can happen via sexual reproduction? Why doesn't sexual reproduction produce clones? Well, but why wouldn't you be able to do that twice and get the same DNA? Combine the same two people's DNA. Shouldn't you get the same thing every time? Shouldn't you get clones? Why? Which part? I've been harping on it. What are the two things in meiosis that mean that cloning doesn't happen from sex? Crossing over and independent assortment. Good. But Ethan's right that every once in a while it happens, right? Twins, identical twins. Now that's because sexual reproduction has already happened, basically. You've got the one cell, then it does mitosis and you get two identical cells, and then you get identical twins. But normally not, right? Plants, though, they're sexually reproducing. There's an out there as well. You can just plant a piece of a plant, right? You can take a leaf from some plants and it'll grow roots and turn into another plant. That's a clone. It's got the same DNA. But for animals, yeah, really the only example is identical twins. That happens naturally. But that's not the kind of cloning we're talking about, right? When we say cloning, we mean taking one cell of an organism and getting it to turn into a whole other organism that has the same DNA. That's like biotech cloning that we're talking about. That's what we're going to need to do with the cat cell that we put the firefly gene into. So I'll teach you how to do that. This is something, by the way, that has been done and done and done at this point. First one was a sheep. You knew that, I think. Following that, we did mice, done dogs. It's really pretty much anything we want to clone at this point, we can. First cloned mammal was a sheep named Dolly. Yeah. Cloning was performed by a Scottish dude named Ian Wilmot. That would be Dolly right there. That would be the sheep that gave birth to Dolly. Is that his mother? We'll have to talk about that. Anyway, basic procedure is this. Actually, let's write it down first. Let's just go through the steps here. How to clone a mammal. Let's say I want to clone. Who are we going to clone? Who are you going to clone? I want to clone. You point to the other guy. It's going to be you. Clone Cole. Do you want to be cloned? Is that what you want? I don't care. I'm going to clone Cole. I'm going to make an army of Coles. I like that. First thing I need is weird, but I need a woman who is crazy enough, first of all, to donate an egg cell. I need a human ovum. And I need a cell from Cole. He's the mammal I want to clone. So I need a cell from him, at least one, probably more than one, that has intact DNA. Then I need an egg cell from a woman. And the first thing I need to do is I need to take a little tiny needle and a really good microscope and a very steady hand. And I need to get the DNA out of that egg cell. I need an empty egg cell for this. We call it enucleating the egg, getting the nucleus out of it. Two ways I can go at that point. I can either shock the two cells to make them fuse, the egg and the Cole cell. I can use electric shock to get their cell membranes to fuse together. Or if I have that steady hand, sometimes it works to suck the nucleus out of the Cole cell and inject it into the empty egg cell. Either way, I'm taking an egg cell that should have a half set of DNA. I'm giving it a full set of Cole's DNA. And I'm basically going to try and trick it into thinking it's a fertilized egg. That's kind of a tricky step. The really tricky step is actually getting it to behave like a fertilized egg, getting it to develop into an embryo. You have to have exactly the right hormones and chemicals that that thing is exposed to. But provided you can get to that step, you're pretty much golden. Because after this, it's something that, you know, obstetricians do thousands of times a day. It's just implanting an embryo into preferably the same woman that donated the egg cell. I get that embryo to implant into that woman's uterus, and I wait 9 months. That's the part science fiction movies kind of skip over. Like, ooh, suddenly we have this army of clones. No, you got to wait 9 months. We don't have this like artificial aging technology. It's a baby, and the baby's got to be born. And now what do I have? Oh, so many questions. So many questions. Is it coal? No, it's not coal. It's like, best I can describe is it's coal's identical twin. 15? You 15? 15 years younger. That's what I've got. But again, so many questions. Like, who are the clone's parents? You? Maybe if you're the one that caused this to happen. But what if I because I don't necessarily have to have his consent to do this. He leaves cells everywhere. I could have just stolen some, cloned him on the fly. Is he in any way responsible for that? Doesn't seem like it. How about the company that paid for it, for this experiment? Are they responsible? Also, generally, when a company pays for a new genetic being like a super corn or whatever, they own that organism. Does the company own Cole's clone? I hope not. I hope we're done owning people. Thought we got rid of that. I mean, there are a lot of questions that come out there. The one that's really going to break your brain is if I had chosen Cole, if I had chosen a female class member, there is the very real possibility that the donated egg could have come from them and that they could have given birth to their own clone. Yeah, it's messed up, right? Okay. So we're there. Change out Cole for a cat, and we can do glow-in-the-dark cat now. Good? I hope you're happy. Soon we will need to start talking about what we should do versus what we can do. First, I want to give you a little more practice with electrophoresis. This is an activity. I want to see how you do. Typically challenging, but I've presented things in a different way, and I hope it makes it easier. This will be due, I think, tomorrow. See if you can do it for tomorrow. Actually, no, let's make it do Friday. So what you've got here is two long lengths of DNA. DNA A and DNA B. Consider them, two different people if you want. And then I'm telling you what recognition site you're looking for, which is the EcoR1 recognition site. And I'm telling you where it cuts. You just got to count how long the pieces are. Now notice I've left out the second strand of DNA. DNA is double-stranded. I'm just showing you the top strand just to avoid confusion. You're showing how many pieces and how long those pieces are, how many bases long they are. And then you're going to show me how far they would go on the electrophoresis gel. You are being given a scale at the top and BP is base pairs, by the way, that a 70 base pair piece would go 2 centimeters, a 50 base pair piece would go 4 centimeters, and so on. Basically, you have to create a scale on your fake gel here that shows you how far each length of piece should go. And then you got to make the bands yourself. Just, draw in the bands. Make sure the bands are the same size and shape as the well that I've drawn at the one side. So get the pattern right and you are good. Questions? See what you can do. Let me know. If it helps, just draw a line where the cut is. I've bolded the recognition sites. You saw that, right? Like you don't have to hunt for them. They're there for you. And now just count the number of letters between each cut. That's how long the pieces are.